no further ado, very pleased to welcome Andrew to talk to us about genomic SEM, which I think is incredibly relevant given all the findings we've seen about genetic correlations and genomic correlations in the last couple of days. Thanks, Andrew.
Awesome. Thanks very much, Talia. So yeah, I'll be talking about some of the new work that we've been doing modeling the genetic relationships across 11 major disorders. And I'm very excited to hear people's thoughts from this particular group and any questions anyone might have. So I just wanted to start by showing this genetic correlation matrix from the most recent cross-disorder paper, which shows the really high level of genetic overlap across all 8 of these disorders. And that's something that's really stood out across all of these talks at the conference, which is this really evolving appreciation for pleiotropy across psychiatric disorders. And so this observation in part led us to develop genomic structural equation modeling, which we introduced in this Nature Human Behavior paper, which in the context of psychiatric disorders can be used to formally model the genetic relationships across the disorders like I just showed you in the genetic correlation matrix. I want to go through now the different iterations of models that we've done for psychiatric disorders. The first being the p-factor model from the original Nature Human Behavior paper, which modeled the genetic relationships across schizophrenia, bipolar, major depression, PTSD, and anxiety, and allowed us to model this kind of common so-called general psychopathology factor. But while we call it a general psychopathology factor here, we really wanted to improve on this model in future iterations by including disorders across a much wider psychiatric space and include much more range of disorders in the model. And we were able to do that in part by partnering up with the cross-disorder group for the Cell paper from this model that Jordan presented on Wednesday, where we modeled 3 correlated factors based on those 8 disorders for a compulsive disorders factor, a psychotic disorders factor, and a neurodevelopmental disorders factor. And we found that this fit the data pretty well for that genetic correlation matrix I showed you at the beginning. Since then, we've updated it still further to include these 3 disorders that I'm showing you here in red, and for alcohol, PTSD, and anxiety. And I want to highlight that even with updated summary statistics relative to the cross-disorder paper for some of these disorders, that we're still pulling out the same compulsive, psychotic, and neurodevelopmental disorders factors. But with the inclusion of PTSD and anxiety, we're now also pulling out this internalizing disorders factor on which MDD loads quite strongly, which I think makes a lot of theoretical sense. And if we look at the genetic correlation matrix across these 11 disorders, what I've done here is ordered the matrix according to the factors that we pulled out in the model. And you'll see that a lot of the factors are really apparent with this ordering. In particular, if you look at factor 4, the internalizing disorders factor, in the lower right-hand portion of the genetic correlation matrix, you can see this really strong subclustering of PTSD, MDD, and anxiety against this overall backdrop of genetic overlap across the 11 disorders. In that factor model I just showed you, there were also some moderate factor correlations across the 4 factors, and that allowed us to also fit this hierarchical factor model where we have the second order P factor that indexes risk across all 11 disorders. Having fit these 2 models, we then wanted to use different sets of analyses to actually evaluate the utility of each of these factors for understanding shared biology or risk factors across these different disorders. And we did that in a number of ways. One of them being that we examined associations with 49 external traits that we deemed relevant to psychiatric disorders. And we were asking 2 questions with these results. One, we were examining what the association was between a given trait X that I'm showing here in green and an individual factor like the compulsive disorders factor. But 2, by comparing the model fit between a factor model in which the trait X only predicted the factor to which it predicted the individual disorders, we're asking to what extent is the association between trait X and the factor representative of the associations at the level of the individual disorders. Or to put it another way, we want to ask whether or not there's a really disorder-specific relationship with trait X, or whether or not it really is shared across all of the disorders. So for these correlation results, I'm going to show them within particular subdomains. And for all of them, they're colored according to this key here at the top for the different correlated factors from the correlated factor model, and for the P factor from the hierarchical factor model. And for bars that are shown with a solid outline, these are relationships that did seem to be well accounted for by the factor, and dashed outlines are relationships that did seem to be highly heterogeneous across the disorders that loaded onto an individual factor based on that model comparison that I just showed you. So for example, within the Townsend Deprivation Index, we see that there's a strong positive association between the neurodevelopmental disorders factor and Townsend deprivation. But if we look at the level of the individual traits, we see that this relationship is primarily driven by ADHD and PTSD, but is non-existent for autism. And so for that reason, this was estimated to be highly heterogeneous. Some homogeneous associations within this subset were a lot of positive genetic correlations between the compulsive disorders factor and different subdomains. And within health and disease, something to notice is a lot of positive genetic correlations between the internalizing disorders factor shown in purple and a lot of these different health and disease outcomes. Finally, within the anthropomorphic domain, I want to highlight the compulsive disorders factors shown here in blue showed a homogeneous relationship with waist to hip ratio and body mass index, which, as Professor Bulick highlighted, this is something that we see within anorexia nervosa for anthropomorphic traits, but it's also something that we observe within OCD and to a lesser extent Tourette syndrome. So it does conform to that factor structure. And then I also just want to highlight these negative associations between the internalizing disorders factor and age at menarche and age at menopause, indicating some sort of association between a generally faster pace of aging and those internalizing disorders. Overall, if we look at kind of the ratio of genetic correlations that were estimated to be highly heterogeneous or not, we see that the compulsive, psychotic, and internalizing disorders factor do a pretty good job of describing the relationships between the individual disorders and these external traits. But for the neurodevelopmental and P factor, we see that most of the relationships are actually obscured by the factor. And so I'm showing those in red because they don't really seem to pass this litmus test as far as the utility of these factors. Using a very similar framework, we looked at the associations between the different factors and individual genetic variants using multivariate GWAS and genomic SEM. I'm going to spend less time on this just for time reasons, but just as a general overview, using a very similar set of model comparisons where we had the SNP predict the factor versus the SNP predicting the disorders, we looked at the number of hits that were homogeneous versus heterogeneous, shown here in the dashed outlines in these bar plots. And what we see similar to external traits is that the psychotic and internalizing disorders factor do a good job of describing the pattern of relationships with individual genetic variants. But once again, the neurodevelopmental disorders factor, and in particular the P factor, really obscured relationships with individual variants, where we only found 2 hits for the P factor and 69 hits that were highly disorder specific or specific to the individual factors. And for the compulsive disorders factor, I would say that the jury is still out right now just because of the lower power of the contributing GWAS for that factor. So I want to end now by talking about some methods development we've been doing and its application to psychiatric traits that ask the question of whether or not there are biological functions that characterize these genetic variants with transdiagnostic or pleiotropic effects. As all of you will be aware, there's a lot of different methods available for examining enrichment within functional annotations, one of the main ones being stratified LD score regression, which is introduced in this Nature Genetics paper in 2015, where they show that schizophrenia and bipolar disorder, like you'd expect, are highly enriched within the central nervous system, shown in orange. And what stratified genomic SEM does is extend this to the multivariate case, so we can examine enrichment of pleiotropic signal at the level of, say, the psychiatric factors that I've showed you here. For different brain cell subtypes, which are going to be the functional annotations I focus on, we see that in general, like you'd expect, psychiatric disorders are expressed in the brain. But with some more recent efforts, including some talks that we've heard in this conference, we're able to look at enrichment of very specific brain cell subtypes. In general, protein truncating variant intolerant genes tend to be enriched in cross disorders, where PI genes are those that are characterized by a particularly low frequency of protein truncating variants, indicating that these might be particularly relevant for overall functioning. So what we, and I should say WONU, did is create annotations for PI genes, brain cell subtypes, and their intersection, and then we examined whether or not there was enrichment for pleiotropic signal within these annotations. And so just to orient you to these results, on the left we have the brain cell types that I just discussed, in the middle the PI genes, and then their intersection on the rightmost part for compulsive disorders factor, followed by psychotic, neurodevelopmental, and internalizing disorders factor. And what we see is that within the psychotic disorders factor, there's general enrichment within the excitatory and GABAergic functional annotations and within the PI annotation, but there is much stronger enrichment within their intersection. So we're getting a much stronger signal And what's really cool is that this is not signal for an individual disorder, but indexes genetic risk sharing across the disorders that load on the psychotic disorders factor. And because this factor is defined primarily by bipolar and schizophrenia, which are highly genetically correlated, I think this gives some real insight into some of the biology that might lead to that genetic overlap across these two disorders. So in conclusion, we've expanded the genetic factor model within Genomic STEM to identify 4 major factors of genetic risk sharing. We see that the psychotic and internalizing factor have relatively high utility based on different litmus tests using correlations with external traits and individual genetic variants. The compulsive disorders factor does a good job with respect to external traits, but maybe needs to have more work done as those GWAS get higher in power on the genetic variant end. And we see really low utility of the neurodevelopmental disorders factor, in particular because autism seems to really stand apart from the other disorders that load on that factor. We can also model a P factor using the genetic correlation matrix, but at the same time, we find that that has really low utility with respect to external correlates, and in particular with respect to individual SNP associations, which we think has some pretty broad implications for what is a rapidly expanding P factor literature, where our sense of things is that we should really be looking for factors that kind of have this utility at different levels of measurement. So while I'm not saying that genetics should stand above other levels of measurement, I think that the fact that these genetic variant relationships are really not holding up for the P factor should give us some pause with how much we kind of move forward with the P factor. And finally, we developed stratified genomic SEM to look at enrichment of pleiotropic pathways. And we find that for some of these brain cell subtypes that are described for schizophrenia, they might be conceptualized more as reflecting broader pathways of risk for these disorders that load on the psychotic disorders factor. And for the particular annotations that we created, we find that the intersection of PI genes, excitatory and GABAergic genes are enriched for this factor, which again, primarily indexes risk, shared risk between bipolar disorder and schizophrenia, I think giving us some real insight into what it is that causes these two disorders to overlap genetically. And so with that, I want to give particular thanks to Elliot Tucker-Drob and Michelle Nabard, who were heavily involved in this project, different people who helped with coding and creating annotations, and individuals who've already given really helpful feedback, and some individual working groups who've provided early access to summary data in order to perform these analyses. And of course, just the PGC, IPSC, and UK Biobank, who all are the provided data or summary data involved in this project. And so with that, I'll just take any questions.
Great, thanks, Andrew. That was a fantastic talk and really exciting to hear all these new developments, and particularly moving into the biology. Perhaps while other people gather their thoughts, I actually just had one quick question, if you wouldn't mind going back to the slide immediately before you moved on to the biology section, where you had the kind of homogeneity and heterogeneity. Yeah, that one. So can I just check I understood what you were saying about the P factor one here? Because to me it made sense that the majority of the genetic variants associated with the P factor were not specific, because that's kind of the whole point of the P factor, is that it's overarching. But maybe I misunderstood what point you were making.
So this is basically showing that we have 2 hits for the P factor, but then we have 69 genetic variants that seem to be really specific to an individual disorder. And so they're popping for this heterogeneity test, or what we call Q-SNPs in genomic STEM. And so these are SNPs that are not operating through the P factor in any way, which you might expect in general. You might expect some specific signal to the disorders, but then at the same time, if the P factor was reflecting any sense of shared biology, you'd still expect at least some homogeneous hits in that leftmost bar for the P factor. So the distinction really is kind of the discrepancy between those 2 bars. Does that make sense?
Yeah, I don't think I'm entirely clear what you're meaning by the heterogeneous ones in that case. How are they associated with the P factor if they're not? I mean, so that, sorry, maybe I'm answering my question very clearly, but so where is their association coming from if it's not coming from the P factor then?
We're basically, if you swap out X here for a SNP, we're basically comparing the model fit where a SNP predicts the P factor to a model in which the SNP predicts the individual disorders or the factor that define.
I get you. Okay, so the majority of them are sitting in that right-hand camp.
Exactly. Yeah, where we see that it really is not describing the relationship very well when we just have the SNP act through P in that way.
Right, despite the fact that the disorders all load onto the P factor.
Exactly, yeah.
Okay, interesting. Thank you for explaining. I hadn't fully understood that, so perhaps everyone else had, but that was very useful for me. Do we have any other questions? Let's have a quick look. Yes, so there's one here. Have you looked at the heritability estimated for the 4 factors you discussed? How do they compare with the heritability estimates for DSM categories?
We haven't looked at the heritability for the 4 factors. We've done overall comparisons of kind of mean chi-square for the factors. And we see that in general, I mean, the point of genomic SEM is not just to get a power boost, but for disorders that do share some aspect of genetic etiology, we do see an overall increase in mean chi-square, but we haven't looked at the heritability yet.